Dear students, welcome to the online class. In this presentation, we will discuss about human red blood cells or RBCs, its number, structure and functions. Blood is composed of 55% plasma and 45% blood cells, also known as blood corpuscles or formed elements. This ratio of 55:45 is known as hematocrit value. The blood corpuscles or formed elements consist of number 1, erythrocytes or red blood cells, also known as RBCs. Number 2, leukocytes or white blood cells, also known as WBCs. And number 3, thrombocytes or platelets. The formation of RBC from bone marrow is known as erythropoiesis. A hormone called erythropoietin secreted by the kidney regulates erythropoiesis, especially during hypoxia, that means low oxygen concentration in tissues. It is also produced in the liver. About the structure of RBC. RBC in man is circular in shape and biconcave disc-like. It is non-nucleated or enucleated, but in all vertebrates except mammals, RBCs are nucleated. It has a diameter of about 7 to 8 microns. The center of RBC has a thickness of 1 micron, but at the periphery it has a thickness of 2.2 microns. RBC is limited by a semi-permeable plasma membrane. Its cytoplasm contains no cell organelles. About 80 to 90% of the cytoplasm of the RBC is filled with hemoglobin molecules. An RBC contains about 270 to 280 millions hemoglobin molecules. About the number of RBCs. The average number of RBCs of an adult man is about 5 millions per cubic millimeter of the blood, and that of a woman is about 4.5 millions per cubic millimeter of the blood. The average lifespan of an RBC is about 120 days, usually between 90 to 120 days. The old cells are destroyed within the spleen or liver. Spleen is considered as the graveyard of RBC. During the destruction of RBC, hemoglobin molecule is broken down and its globin or protein portion is taken back for the resynthesis of a new hemoglobin molecule. A hemoglobin molecule consists of a coloring portion called heme and a protein portion called globin. Heme consists of an iron porphyrin ring. Each ion of the heme can combine with one oxygen molecule. Since there are 4 heme in a single hemoglobin molecule, each hemoglobin molecule can carry 4 oxygen molecules. The globin or protein portion consists of 4 polypeptide chains. 2 chains are identical, known as alpha chains, and other 2 chains are identical, known as beta chains. Each alpha chain has about 141 amino acids and each beta chain has about 146 amino acids. During the destruction of RBC, the heme portion is eliminated as a waste except its iron molecule, which can be reused for the synthesis of a new hemoglobin molecule. the porphyrin portion of the heme is metabolized by the liver and converted into yellow pigment called bilirubin, which is a waste, hence eliminated out through the fecal matter. An abnormal increase in the number of RBC is known as polycythemia. Several factors can stimulate polycythemia. They are high altitude, vigorous muscular exercise, high temperature, emotions, etc. During certain lung disorders, the low oxygen content, that means low PO2 of the blood, may stimulate the excess production of RBCs. The abnormal increase in the number of RBCs can result in the increased viscosity of the blood, which in turn may lead to the blockage of blood vessels. An abnormal decrease in the number of RBCs is termed as erythropenia, also known as erythrocytopenia. One of the adverse effects of erythropenia is that it can lead to an abnormal decrease in the hemoglobin content of the blood. This condition is known as anemia. About the functions of RBCs. RBCs help to transport the oxygen from the lungs to tissues. About 25% of the carbon dioxide is transported from the tissues to the lungs with the help of RBCs. The hemoglobin of the RBC helps to maintain an acid-base balance of the blood through its buffering action. RBCs also help to keep the normal viscosity of the blood, which is an essential factor for regulating the normal blood pressure. These are some of the basic information about human red blood cells. Thank you for watching this presentation. An RBC contains about 270 to 280 million hemoglobin molecules. An RBC contains about 270 to 280 million hemoglobin molecules. An RBC contains about 270 to 280 million hemoglobin molecules.